Welcome to our first semester cell biology course. Today, we're going to explore one of the most fundamental processes that keeps every living cell alive: how molecules travel across the cell membrane. Imagine a city wall with gates that must constantly let supplies in and waste out. The cell membrane does exactly this, using two main strategies, known as active and passive transport. Understanding these mechanisms is absolutely essential for grasping how life functions at the microscopic level. Let us begin with passive transport. Imagine you are at the top of a steep hill on a bicycle. If you take your feet off the pedals, you will naturally roll down to the bottom without doing any work at all. This effortless movement is exactly how passive transport works in a cell. Molecules naturally want to spread out from crowded areas to less crowded areas, just like people leaving a packed concert hall to step into an empty street. The cell does not have to spend any of its precious energy to make this happen. It simply opens a door and the molecules flow down their natural gradient. Now that we understand the analogy, let us look at the scientific mechanism. In scientific terms, passive transport is driven entirely by the kinetic energy of the molecules and the existence of a concentration gradient. Molecules will spontaneously move from a region of higher concentration to a region of lower concentration until equilibrium is reached. This includes simple diffusion, where small, uncharged molecules slip right through the membrane, and facilitated diffusion, where larger or charged molecules use special protein channels as doorways. Without this free process, life would cease to exist. Every time you take a breath, oxygen naturally diffuses into your blood, and carbon dioxide diffuses out, entirely for free. This natural movement we just discussed can be calculated using Fick's first law of diffusion. Conceptually, this formula tells us exactly how fast molecules will flow across a barrier. Let us break down the symbols. J represents the diffusion flux, which is the amount of substance moving through a specific area per unit of time. D stands for the diffusion coefficient, telling us how easily a specific molecule moves through the medium. Delta C is the concentration difference, and delta X is the membrane thickness. The negative sign is crucial here. It shows that movement happens in the direction of decreasing concentration, from high to low. If the concentration difference increases, the rate of diffusion will also increase, meaning a steeper gradient causes faster movement. Now that we have our formula, let us apply it to a real cell. We are given a cell membrane with a thickness of 10 times 10 to the negative 9 meters. The oxygen concentration outside is 0.5 moles per cubic meter, and inside it is 0.1. We need to find the diffusion flux, given a specific diffusion coefficient. First, we calculate the change in concentration by subtracting the outside concentration from the inside, giving us negative 0.4. This negative tells us oxygen wants to move inward. Next, we plug our values into Fick's law. Notice how the negative sign from the formula and the negative from our concentration difference cancel out. Performing the math, we find that 4 times 10 to the negative 4 moles of oxygen flow through every square meter every second. This positive value confirms oxygen is moving inward to keep the cell alive. Let us visualize the process we just calculated. Here, you are looking at a cross-section of a cell membrane. Notice the extracellular space at the top, which is packed tightly with blue molecules, representing a high concentration. In contrast, the intracellular space at the bottom has very few of these molecules. The large blue arrow pointing downward represents the passive transport, or diffusion. It clearly shows the spontaneous flow of molecules from the crowded area to the less crowded area. The cell does not provide any energy here. The molecules are simply following the rules of physics, slipping through the membrane to reach equilibrium. Having seen how molecules flow naturally, we must now ask, what happens when a cell needs to move things in the opposite direction? Imagine you are at the bottom of that same steep hill, but this time you need to push a heavy boulder all the way to the top. The boulder will never roll up the hill on its own. You have to sweat, strain, and burn calories to force it up. This is the perfect analogy for active transport in a cell. Sometimes a cell needs to stockpile nutrients that are already highly concentrated inside, or it needs to pump out toxins that are highly concentrated outside. To move molecules against their natural flow, the cell must step in and pay a steep energy price. Building on that analogy, let us examine the biological machinery that makes this uphill push possible. Active transport is the cellular process of moving molecules or ions across a cell membrane from a region of lower concentration to a region of higher concentration. Because this movement goes against the natural concentration gradient, it requires the expenditure of cellular energy, almost always in the form of ATP. Special transmembrane proteins act as molecular pumps. They bind to the target molecules and physically change their own shape to push the molecules across the barrier. There are two main types: primary active transport, which directly burns ATP, and secondary active transport, which cleverly uses the energy stored in an existing gradient to drag another molecule along for the ride. Now that we know how these pumps work, we must understand why they are so vital. Active transport is absolutely critical for maintaining the delicate internal environment of the cell, a state known as homeostasis. For example, your nerve cells rely on the sodium-potassium pump to constantly push sodium out and pull potassium in. This creates an electrical charge across the membrane. Without this active pumping, your brain could not send a single thought, and your heart muscles could not contract. It is also exactly how your intestines manage to absorb every last drop of glucose from your food, even when your blood already has plenty of sugar. To understand exactly how much energy the cell must spend, we use the free energy change formula. Conceptually, this equation calculates the total energy required to force a molecule across the membrane against its will. Let us define the terms. Delta G is the total free energy change. R is the universal gas constant, and T is the absolute temperature in Kelvin. The natural logarithm term accounts for the chemical gradient, comparing the inside and outside concentrations. The second half of the equation, ZF delta psi, applies if the molecule has an electrical charge, factoring in the voltage difference across the membrane. If delta G is positive, it means the process requires an input of energy, perfectly describing active transport. As the inside concentration increases, the energy required shoots up significantly. Let us put this formula to work with a concrete example. We want to calculate the energy required to pump an uncharged molecule into a cell at human body temperature, which is 310 Kelvin. The inside concentration is 100 millimolar, and the outside is 10 millimolar. Because the molecule is uncharged, the charge variable Z is 0, meaning we can completely ignore the electrical part of the equation. We substitute our gas constant, temperature, and the concentration ratio into the simplified chemical gradient formula. The ratio of 100 over 10 simplifies to 10, meaning the cell is pumping into an area 10 times more concentrated. Multiplying the constants gives us 2,577.34, and the natural log of 10 is approximately 2.303. Multiplying these yields our final answer, a positive 5,935.6 joules per mole. This positive value confirms the cell must burn ATP to provide this massive energy input. Let us look at the physical machinery that performs the hard work we just calculated. In this diagram, you see the cell membrane with a specialized transmembrane protein acting as a pump. Notice the red square molecules. There are very few in the extracellular space at the top, but a massive stockpile in the intracellular space at the bottom. The pump is actively forcing more red squares upward from low to high concentration. Crucially, look at the energy symbol next to the pump. It shows ATP being broken down into ADP and phosphate. This visualizes the exact energy expenditure, the thousands of joules we just calculated being burned to push those molecules against the natural flow of the universe. We have covered a tremendous amount of ground today, so let us summarize our key takeaways. First, we learned that passive transport is a free, spontaneous process where molecules flow down their concentration gradient, much like rolling down a hill. We used Fick's law to calculate exactly how fast this natural diffusion occurs. Second, we discovered that active transport is the cell's way of fighting against the natural flow of the universe. By burning energy, usually as ATP, molecular pumps force molecules from low to high concentration to maintain vital homeostasis. Finally, we used the free energy equation to calculate the exact energy cost of this hard work. You now understand the fundamental rules that govern how every cell interacts with its environment. Excellent work today.